Hey everybody, we are looking at section 15.2, which will kind of wrap up our content for our chapters 12 and 15 tests coming up here. So for section 15.2, our title is Human Genetic Disorders, and as with 15.1, we are really focusing here on this, in this chapter, on human-specific traits and human-specific inheritance. So go ahead and get the title written down. And what we're looking at here on this slide is a picture of some red blood cells that are misshapen. And this misshapen kind of structure of these red blood cells is representative of a genetic disorder called sickle cell anemia. We'll talk about exactly what that is here as part of this section. All right. So go ahead and get, again, the title jotted down and we'll move on. So when we look at this section, we are going to talk first of all about, I guess, two different types of disorders and where they can come from. So we're going to talk about chromosomal disorders first, those types of genetic issues that come from errors in meiosis where either chromosomes didn't separate properly or cross over properly and so on. So those same types of errors that we looked at a little bit in 15.1 with karyotypes. And then we're going to look at how mutations in DNA specifically in the genes that we hold in our chromosomes can impact the expression of phenotypes and human traits. All right. So this first one here, non-disjunction, is a new term. We haven't really talked about that before, but what we're referring to when we say non-disjunction, and it's kind of interesting, it's almost one of those double negatives. But if I say something is disjointed, the idea is that it is, if it's disjointed, it means that it's disconnected, I should say. So if it's not disjointed, that means that it is connected. So it's kind of a way of just saying that our chromosomes never fully separated, they remained connected. So non-disjunction happens when specifically our homologous pairs fail to separate in meiosis 1. So this can happen either with pairs of autosomes or with pairs of sex chromosomes, specifically X's are what we're thinking of. So in females, those X chromosomes, not so much involving the Y, okay, because the X and Y are not homologous and they really don't pair up. But only for those homologous chromosomes, so 1 through 22, and then X's in females. If those don't separate properly in anaphase 1, when they're supposed to kind of be pulled apart, we can end up with cells that have an abnormal number of chromosomes. And so the trisomies and the monosomies that we talked about in 15.1 are actually derived from this process. All right, so you can see I have a normal cell here with two pairs of homologous chromosomes. I've got the red pair and the yellow pair. If the yellow pair don't separate properly, I would end up with a cell here that has both yellow chromosomes and one red, and then just one red chromosome here. So the red chromosomes separated the right way, the yellows did not. And as a result, when we pull the sisters apart, these two cells on this side are going to have two additional chromosomes, right? Actually, just one. We were supposed to have one of each of these yellows and these. But it's almost like double the amount as what we were supposed to have. This is where we get gametes that have too many chromosomes or too few chromosomes. And then if those gametes are used for reproduction, that's where we end up with individuals that have trisomies or monosomies. So like Down syndrome or Turner syndrome, which is where you're lacking an X chromosome, both of those come back to the process of meiosis as it occurred in the parents, okay? So when we look at these chromosomal disorders, again, we can look at karyotypes, and this is another example of a karyotype. And towards the bottom, they're highlighting for you the mutation that we can see, which is trisomy 21, also known as Down syndromes, okay? So probably the most commonly seen trisomy and most survivable trisomy that's out there at this point. Turner syndrome is what I mentioned, is where you're lacking an X chromosome. You only have one X, you don't have a Y, so here you would be lacking one of these two X's. Klinefelter's is where you have XXY, and for those individuals, they're going to be kind of similar to what females do normally, which is to turn off one of their X's, and they're also going to express the Y that's present. So for all intents and purposes, they'll look like males. Their sex will be males. But oftentimes for individuals that have abnormal numbers of chromosomes, have monosomies or trisomies like this one, they're not going to be able to reproduce, okay? And again, all of this comes back to this non-disjunction process that happens in meiosis, okay? Most trisomies, most monosomies, aka aneuploidies, the word that we used in 15.1, are going to be lethal, which means that these individuals don't typically survive even to be born. But in the case of Down syndrome and Turner syndrome and Klinefelters, they do survive. But again, that's because we're dealing with kind of unique situations here with the X chromosome and then very small chromosome 21 down here at the bottom. Okay. Now, chromosomal disorders are super easy to identify with karyotypes, but what's not as simple is to identify gene disorders. So mutations or changes that occur to DNA sequences that lead to changes in a phenotype in a way that can be detrimental. All right, so when we mutate a gene, essentially what we mean is we're kind of changing the coding for that gene. So maybe, we thought about before, the gene is kind of like your recipe, your ingredients for making a protein product. In this case, if we're changing our recipe in an unproductive way, we're not going to get the right protein product as a result. So when we change them, we have some different ways that our cells can do that. And ideally, none of these changes happen anyway. But we know, and we'll talk about this in chapters 13 and 14, that these changes do happen and they're kind of inevitable. So the way that a DNA sequence can be changed or mutated in the context of a gene and a phenotype would be to change the amino acid sequences. And sometimes that happens as a result of what's happening while we're reading off a gene, while we're reading off the recipe to make our protein. Okay. They can also change a protein by accidentally kind of increasing or decreasing the protein size. Regardless of what the impact is, it's going to usually cause some type of disorder and specific phenotypic consequences. So the example of sickle cell that I showed you before, this same picture represents one single letter change to a gene that is hundreds of letters long. And with that one single letter change, we get red blood cells that are supposed to be kind of shaped like this. Remember these biconcave discs? They're going to be changed to be sickled in shape, right? And this certainly does not look like a normally shaped red blood cell. And that'll lead to a whole lot of issues for anybody that has red blood cells naturally that are being produced like that. So small individual changes in a gene can lead to like huge impacts on the ability for cells and proteins to function properly, okay? Now, what's helpful about these gene disorders for you is that Mendel's rules of inheritance still apply here. All right, at least if we're talking about an autosomal type of trait, right? Mendel's rules will still apply to autosomal traits, just like we've seen in the past. So again, those are chromosomes 1 through 22. These are those chromosomes that have nothing to do with the sex of an individual, right? These are those traits that are normal body traits and that are present in both males and females. So when we think about normal autosomal disorders, remember we have 22 pairs of these things. Both males and females have 22 pairs. So as with everything we've looked at so far with Punnett squares, we're still going to inherit 2 copies of these alleles. But what that means is that we have a scenario here in a normal dominant and recessive kind of Mendelian pattern. We have a scenario here where we can have heterozygous individuals that have the allele for the disorder, but they're not actually going to show it. And so the term that we use for those individuals is to say that we have carriers, okay? The idea is to say that, okay, you have a carrier individual, a parent perhaps, that has the allele, doesn't show it, so you can't just look at them and see that they have it, but they have it, and they can potentially pass it on to their offspring, right? So if we're talking about an individual that's heterozygous, well, the chances of passing that down to your offspring, right, are 50%. If this is my disorder allele and that parent has it, well, this parent's going to pass it on 50% of the time to this individual or to this individual. Okay, and then the same with this parent, right? They're going to pass it on 50% of the time as well. And so when we think about this, those carriers become really, really important. All right, so we don't just use the word heterozygous this time. We call them carriers if possible. All right. Most disorders are going to be caused by recessive alleles, though there are some examples of some that are dominant. All right, so if we look at some examples here, autosomal recessive disorders include things like cystic fibrosis, which is a It's a disorder that has to do with difficulty in breathing. All right, so there's a small change in a chloride ion transporter in the cells that make up your lungs. All right, and so for someone who has cystic fibrosis, they have that mutation. That transport protein no longer functions properly, and those ions can't be moved across the membrane. And it sounds like such a teeny tiny little problem to have, except for if we can't transport those things across the membrane for whatever reason, mucus starts to build up in the lungs and in the windpipe. And what that means is that if you can imagine, if you've ever been sick and kind of that drainage, right? Or if you've ever had something like pneumonia where you've actually got fluid in your lungs and you cough and you kind of hear it in that cough, people that have cystic fibrosis, that's kind of a constant battle for them. So they have to continuously get rid of and expel this mucus in order to remain able to breathe. It can be really kind of scary and life-threatening, okay? And that is, again, autosomal and recessive, which means for anybody that's going to inherit that, they have to have gotten it from both mom and dad. So meaning both parents had to have been carriers, okay? Galactosemia, phenylketonuria, and alkaptonuria are all disorders that have to do with an inability to digest something. All right, so galactosemia is an inability to digest something called galactose, which probably sounds an awful lot like lactose. It's also a sugar. It's a type of carbohydrate, and if they can't break it down, it's a little bit more severe than lactose intolerance, but there are certain foods and things that these people would need to stay away from. Same with phenylketonuria, which is often called PKU. There's a lot of processed foods that near the nutrition label will say things like, it references phenylketonurics, which is those types of compounds that people with PKU kind of have to stay away from. Albinism is what you've heard of, no doubt, before, which is an albino individual. And those individuals inherit, again, 2 copies of an autosomal recessive allele. Now, what's interesting about albinism is kind of this extra powerful disorder in a way. Remember back in 12.3, I talked about the fact that when we talk about skin color in humans, there are many genes that work together to influence skin color. But what we also know is that when you have albinism, it's just 2 copies of one gene that kind of covers everything else up. So albinism, again, in order to inherit this, you had to have 2 parents that were both carriers of that trait or were affected, and then they passed it down to you. Okay, dominant disorders, Huntington's disease is a kind of a progressive neurological disorder where brain tissue, nervous system tissue starts to break down and it becomes difficult to kind of maintain just normal daily tasks. Achondroplasia is dwarfism. That's, like little people. That's also dominant, which is kind of interesting because that means that 2 individuals that are dwarfs actually could have a normal sized child, right? If they just had 1 copy of that dominant allele. And then polydactyly is where you have extra fingers and extra toes, right? Like I don't have extra fingers or toes, but it would be like individuals that sometimes have 6 and they can have like a little projection, growing off the side there. Okay, also dominant. With Huntington's, achondroplasia, and polydactyly, these aren't necessarily always things that we inherit, but these are small changes in DNA that can occur at random in the production of gametes that then can be passed down. So it's not like every parent in the line of someone who has extra fingers and toes also had extra fingers and toes. There can be random changes that happen that are a little bit more unpredictable, okay? All right, so looking at gene disorders that are not autosomal, and I've just got this slide, and then we're going to work on some Punnett squares, okay, to kind of start using some of these terms. There are sex-linked or X-linked genetic disorders, and they can be recessive sex-linked, and they can be dominant sex-linked, but the majority of anything on the X that is similar to a disorder is going to be X-linked and recessive. So red-green color blindness is one example. This is where, again, if you inherit on the X chromosome that color blindness allele, you tend to be colorblind all the time if you are a male. And then females, because they have 2 X's, can kind of express that like we would express anything autosomal. And we'll look at that with the Punnett squares here in a second, okay? Hemophilia is another example of something that's X-linked and recessive. And again, as with color blindness, males tend to express that much more often than females. Hemophilia is a bleeding disorder, and the picture you see here on the right is of exactly what happens in, let's see here, a normal individual and an individual that has hemophilia. And so the difference being, in normal individuals, we have the ability to clot our blood. We have platelets and fibrin and other types of small proteins that help to kind of coagulate red blood cells if you have a cut, and hold them in place to allow you to form scabs and then heal that wound. People that are hemophiliacs don't have those activated platelets and fibrins and other proteins that are necessary to clot blood. And so even a very small cut, right, like hypothetically you fall off your bike and you scrape your knee, or you get a pretty good paper cut or something, that would run the risk of actually causing someone to die just simply through blood loss, because their blood just simply does not stop. And people right now that have hemophilia have the ability to go in and get treatments for these things, but it's still have to be really cautious when they're undergoing things like surgery if they have hemophilia. Most sex-linked disorders, as I mentioned, are going to disproportionately affect males, so meaning more males are going to have the disorder than females. Females, again, express our sex-linked or X-linked disorders just like we express anything on our X, in that we have 2 copies. And with those 2 copies, whatever's dominant will show. Recessive can be covered up if there's a dominant allele present. Now, that being said, because females can be heterozygous and have dominant and recessive, if a disorder is recessive on the X chromosome, females can carry those. Females can be carriers and pass them on to their offspring. Males, on the other hand, can never carry an X-linked trait or disorder without showing it in the phenotype. So males, it's either they have it or they don't, and it's very much cut and dry. Females, on the other hand, there can be some questions sometimes about whether or not she could be a carrier or if she's, potentially homozygous. Okay. So I want to do a couple examples of Punnett square. I'm going to try and do them here on the slide with you. So we're going to write down a couple of examples, and of course, anything I put up on the screen, I want you to add to your notes also. So if we think about autosomal recessive disorders, I want to stick with one of those that we talked about on the slide just a few slides ago, I should say. So that's going to be cystic fibrosis. All right, and so with cystic fibrosis, again, it's autosomal, so that means we can just use regular letters. We don't have to attach anything to an X. And it's also going to be recessive. Okay, and remember, with any recessive trait like this, that we're going to write our letters a little letter and make that kind of count as a disorder in this case. So here we're going to say, I'll just use A's for simplicity. So we'll say big A represents normal, unaffected, no disorder. So I'm going to put no disorder. You could put any of those other ones if you'd rather. So unaffected, if you want, would be good. And then little a in this case is going to represent cystic fibrosis, or you could say disorder. Oh, and I'm going to be out of space here. Hold on, let me come back. Cystic fibrosis. So we'll just say that's good there. So if I take 2 individuals, let's just say I want to cross big A, little a with big A, right? And those are my parents. Here again, because it's autosomal, it does not matter which one is male and which one's female. We're just using A's. But if we think about how we could characterize these parents, right? This parent over here has no disorder and is normal. Okay, also is not a carrier, but this parent over here is a carrier. This parent over here doesn't actually have cystic fibrosis and would live a pretty normal life, except for the fact that they could pass down that little a allele to their offspring, okay, and potentially not even know it. So if we put, big A, this should have gone here, big A, little a, and cross it with big A, okay? So we set it up just like we've always set our Punnett squares up. So big A, little a, big A, big A. And if I do that cross, it's just like what we've seen before. I'm going to get a couple homozygous dominant, and I'm going to get a couple that are heterozygous. So my genotype ratio, I could say I have 2 big A, to 2 big A little a, or 2 homozygous dominant to 2 heterozygous, or 2 homozygous dominant to 2 carriers. Phenotypes, we're going to say all or 100%, all unaffected, all no disorder, all normal. And I kind of hate to use that word normal with this because you're still normal regardless, but we're saying normal in the context of you do not have whatever this disorder is, okay? So if you think about something that's autosomal recessive like this, to have a disorder would mean that you have to inherit 2 of these little alleles, okay? Which means both parents would have at least had to have been carriers. Now this is going to look different when I flip over and we look at autosomal dominant, all right? So let's do that. If we consider an autosomal dominant disorder, let's use dwarfism as our example, right? Little people. So when we talk about dwarfism, this is again autosomal, so we just need to use a letter, right? Not attached to an X or anything else, but it's dominant in this case. So with a dominant disorder, we would say we could still use the same letters if we want, or you could change it to D's or whatever you want to use for this. I'm going to stick with my A's. But now my big A here is going to represent dwarfism or disorder. And my little a in this case is going to represent that they're unaffected or no disorder. And so when I go on to do this cross, if I cross the same 2 parents, I'm going to go big A, little a, cross with big A, big A. Well, in this case, if we think about our parents, this parent here on the right now, instead of being homozygous and not having anything to do with the disorder, this parent now is homozygous for the disorder. So this parent has a disorder. This parent would actually be considered a dwarf or a little person, and is definitely going to pass that down. This parent, on the other hand, only has 1 copy of that dominant disorder allele. However, we can't cover up a dominant disorder allele. So that parent also has dwarfism, all right? But she or he also has the little a, right? We do not call this parent, in this case, a carrier, because you cannot carry a dominant allele. If you have it, you show it. Okay, end of story. You don't carry a normal allele. We don't talk about it like that. We only use the term carrier in the context of carrying a carrying a disorder allele without actually showing it. Okay, so if I cross these 2 parents, so big A, little a, big A, big A, I'm going to get the same phenotype, I'm going to get the same genotype ratio, but my phenotypes in this case are actually going to differ. So for my genotypes here, I could again say I have 1 big A to 1 big A, little a, or 2 to 2, or 50% to 50%, or half to half. All of that would be equivalent. For my phenotypes, we're going to say all affected this time. And they're all affected this time because, again, we're dealing with a dominant disorder, right? So all that you need is 1 dominant allele, and every single one of these has at least 1 copy of a dominant allele. So notice how that's different from what we saw on the last slide, right? The last slide, all were unaffected, and here they're all affected. So the Punnett squares, again, are exactly the same. What's differing this time, again, is how we're interpreting our phenotypes. So when you see these types of questions on a quiz or on a test, right, pay attention again, as I say every other day, I swear. Pay attention to what's being asked for, and think about that in the context of what you've learned, okay? And it certainly wouldn't hurt to have a couple of these jotted down as examples on your notes page, okay? So the last Punnett square I want to talk you through is going to be the one for a sex-linked recessive disorder. So if you remember what we did in the past, all right, sex-linked traits of any type are always going to be written on the X chromosome because that's where we find them, right? Sex-linked is the same as saying X-linked. All right, so for an X-linked disorder, it's going to be actually like physically attached to the X, all right? Let's say for this one, we want to do hemophilia, okay? So the bleeding disorder where your blood cannot clot. All right, now for hemophilia, what I want to do is I'm going to label this again attached to the X. And again, it's recessive. We're going to assume if it's X-linked, it's always going to be recessive because that's far more often going to be the case. So I'm going to write it like this. I'm going to say X with a big H is going to mean that we have an individual that has no disorder, or you could say, again, unaffected or normal, okay? In contrast to that, we're going to say X with a little h is going to represent hemophilia or disorder or affected. Again, any of those would be just fine. In this case, I'm going to cross two parents, and again, I have to keep in mind here I'm dealing with a male and a female, and gender actually matters in this case. So for my first parent, let's do the male here, X big H and Y. That'll be his genotype. And then for the female, we're going to say X big H and X little h. Okay, so this individual here on the left, the male, we could say is a few things. He's male because he's got a Y. And secondly, he has no disorder because the only X chromosome he has just a big H on it, which again codes for no disorder. This person, on the other hand, again, female because she's got two X's, but now she actually has a little H allele. Does she show it though? No, because she's got that dominant big H there to kind of cover it up. So in this case, we call her a carrier because she has it, but she doesn't show it, okay? This is the exact same reason why a male cannot carry one of these traits without showing it, because again, he only has one X. Whatever's there is always going to be expressed, okay? So if I set up my cross, we'll do the male here and the female on the side. And what we should get here is a variety of genotypes and phenotypes, really. So let's see, X big H, X little h. Let's see if I can fix that. There we go. X little h and Y. Okay, so in reality, I actually have 4 different types of genotypes, right? I have X big H, X big H. I have X big H, X little h. Those are both of my females. And then my males, I have one of each of those also. All right, so instead of trying to write all the genotypes on that line, which I could, but I would essentially just do one of these, 1 to 1 to 1 to 1, right? 25% chance of each. I'm going to also go in here and describe them on this side. Okay, so let's say here I want to talk about this individual here first. So this person, I know I can say this person is a female because she has 2 X's. Whoa, come on back. So I know she's a female because she's got 2 X's there. I also know she is not a carrier, right? So no disorder, not a carrier. And you could have also said right there, she's completely unaffected, right? If I go on here to, let's say, this individual. Okay, here's my other female. This female here, so I can also label again female. This female here, though, on the other hand, has a big H and a little h, kind of like mom did. So again, we can call her a carrier, okay? This individual over here is not a female at all. This person has a Y. So this would be male and no disorder or unaffected, right? He's definitely not a carrier. He doesn't have the little allele. He just has the one big H allele. And then for my very last individual here, X little hy, this is a male who is affected, right? So male, because he's got a Y, and hemophilia. So what this means is that on a test, on a quiz, I could have you set up a cross like this. I could even give you your parent genotypes. And I may ask you, what's the probability of a female being produced that has hemophilia? And your answer would be to say 0%, because based on this cross, neither of my females over here are actually going to be hemophiliacs, right? They don't have the disorder. One of them is a carrier, but neither of them actually expresses it, right? I could also say, what's the probability of producing a male who is a carrier? And your answer for that will always be 0, because males can't carry these traits, right? However, if I said, what's the likelihood of producing a male that is unaffected or is normal, you could say 50%, right? Because of the males, that would be 50%. All right, so you've got to be able to kind of separate these two, right? Females versus males, and keep those straight because, again, those X's and Y's are really important here with this. All right. So the very last thing I want to talk about was a really cool interaction here between the environment and also genotype. All right. So sickle cell anemia, I mentioned earlier, it is what happens here, all right, to create red blood cells that look misshapen like that. When we talk about sickle cell, what's really kind of interesting about that is sickle cell allows for some protection of things. There's a reason why it's still around. So sickle cell, although it misshapens and kind of changes your red blood cells in a detrimental way, in a way that makes your red blood cells have more difficulty to try and move oxygen through the bloodstream, it does also help prevent diseases like malaria. So what we've seen happen, malaria here is endemic. It means that it's, pretty much pervasive. It's here all the time in this part of the world, especially in these kind of subtropical areas near the equator, also in South America, by the way. But Africa is kind of the area where we struggle with it the most. And malaria has been there for a very long time. Now, malaria is transmitted by mosquitoes. And what we've seen happen, what scientists have been able to identify, is that this sickle cell allele, which is this HBS allele that's being labeled right here, that the more commonly we find that allele, the more that kind of overlaps with the spread of malaria. And what they've identified is this idea that misshapen red blood cell is going to kind of prevent the malaria parasite from infecting those cells. So malaria normally infects your red blood cells and will attack your body that way, and it almost always becomes fatal and lethal. But if you carry a copy of that sickle cell allele, you're essentially kind of immune to malaria. And so over time, as these populations have kind of spread throughout the world, right, the sickle cell allele has also kind of spread. The danger would be if you inherit 2 copies of that allele and all of your red blood cells become misshapen, then you're really going to struggle in terms of actually being able to move oxygen well. And then you will have some other serious kind of more genetic issues, okay? You'll still be protected from malaria, but inheriting 2 copies is also going to be dangerous in that regard, okay? Same thing about this. CFTR, by the way, is the allele that is mutated when someone has cystic fibrosis, okay? So cystic fibrosis transporter is what that stands for. We've also identified that if you carry 1 copy of that allele, that mutated gene that normally causes cystic fibrosis, if you are a carrier of that, then that protects you from typhoid fever, which is caused by a bacterial cell that can make you very, very ill. And so for that reason, that's why we tend to see these alleles continue in a population, even though something like sickle cell and cystic fibrosis can cause early death. There are individuals that are surviving with this because they're protected from these other types of transmissible diseases, right? Not genetic, but transmissible, like malaria and typhoid fever. Okay, pretty cool. So that's all I have for 15.2. We will do some more practice with these Punnett squares and talk more about disorders and how they relate to the stuff from 15.1 and pedigrees. But beyond that, if you have any other questions, let me know. Otherwise, I will see you for the next chapter when we get there. All right. Bye, y'all.